Hello everyone, I am Miss Priyanka Sanjay Latte, working as lecturer at More Education Society's Mura Rural Institute of Pharmacy Sone. In this presentation, I am going to discuss topic that is antimalarials. This topic we will discuss for first year D Pharmacy students. Content of presentation: Introduction, Definition of antimalarials, Synonym, Biological source, Geographical distribution, Organoleptic characters, Chemical constituent, Cultivation and collection, Microscopic characters, Chemical taste, Uses and substitutes of following drugs. In that first one, CINCHONA. Now firstly we will discuss introduction. Malaria is an infectious disease caused by 4 species of protozoan Plasmodium. It gets infected into human body due to bites of female of certain species of Anopheles mosquitoes. The drugs used for the treatment of malaria from herbal origin are Artemisia annua and CINCHONA. Next point, definition of antimalarials. The drugs which are used for the prophylaxis or treatment of malaria are called as antimalarials. Example, CINCHONA, Artemisia annua. Next point, we will discuss the core drugs. CINCHONA. Synonyms: justice bark, premium barks. Biological source: it consists of dried bark of trees of CINCHONA calycea, CINCHONA legergenia, CINCHONA succirubra, CINCHONA officinalis belonging to family Rubiaceae. Geographical source: the countries producing the drug on commercial scale are Tanzania, Bolivia, Colombia, Ecuador, Guatemala, Indonesia, Zaire, Kenya, Sri Lanka and India. At present, most of the world requirement is met by Zaire and Indonesia. Approximately 5,000 tons of CINCHONA bark is processed annually to produce alkaloids all over the world. In India, it is being cultivated in Tamil Nadu, West Bengal, Karnataka, Assam and Sikkim. Approximately 5,000 hectares of the land is under cultivation of CINCHONA in India, of which about 2,500 hectares is in Tamil Nadu. In India, CINCHONA is cultivated in Antimalarials Hills in bracket Coimbatore district bracket complete and Nilgiri Hills in bracket Nilgiri district bracket complete in Tamil Nadu. It is also cultivated in Darjeeling district of West Bengal. The government factories extracting CINCHONA alkaloids are situated at Mungupo in bracket West Bengal bracket complete and Antimalarials in Tamil Nadu. Next point, cultivation and collection. Most of the CINCHONA species thrive best in tropical climate at an altitudes of 1,000 to 3,000 meters. The soil temperature, rainfall and other requirements of CINCHONA plant are as under. First one, altitude 1000 to 3000 meters above sea level. It also grows satisfactory below 1000 meters, but it has been found to contain low quinine. Second one, temperature. The optimum temperature for the favorable growth of CINCHONA is 60 degrees Celsius to 75 degrees Celsius Fahrenheit. Third one, rainfall in between 250 to 380 centimeters, well distributed throughout the year. Fourth one, soil. Light, well-drained forest soil rich in organic matter is found to be suitable for CINCHONA cultivation. The acidic soil with a pH of 4.2 to 5.6 and a little nitrogen content is found conducive to the maximum growth of CINCHONA. High humidity, sloping situation, shelter from wind are also necessary for CINCHONA plantation. Cultivation of CINCHONA trees can either be done by sowing the seeds or budding and layering. In Tamil Nadu, the budding and layering methods of vegetative propagations are followed, while in West Bengal, only budding is followed. The seeds of CINCHONA are very small and light and about 3500 seeds weight only 1 gram. The seeds are supplemented with sand to keep them in position while sowing. It takes about 3 to 6 weeks for their germination in the nursery beds. The seeds are sown as early as possible after their collection as they lose the variability on storage. The seedlings, which bear about 2 pairs of leaves, are transplanted in the nursery beds. The space of 6 to 10 cm is maintained between 2 seedlings and 2 rows. The seedlings are protected from direct sunlight. The young seedlings thus produced are transplanted into the forest soil after 14 to 15 months before it rains heavily. The distance maintained in between two plants is roughly 2 into 2 meter. The artificial roof provided to the plant is gradually removed. Since CINCHONA consists of stem as well as root bark, the age of the plant selected for harvesting the bark varies from 4 to 20 years. Maximum alkaloidal content is found in 10 to 12 years old plants. The techniques of coppicing is followed for collection of bark. The vertical incisions are made on the branches as well as trunks of these trees with copper sleeves, and these incisions are connected by horizontal circles. The bark is then removed and dried mostly. Drying of bark is done by natural method, or if done artificially, it is dried below 175 Fahrenheit. Bark loses about 70% of its weight on drying. The dried barks are then packed in gunny bags and marketed. Next point, organoleptic characters. Color, stem bark. Outer surface is dull brownish grey to white. The inner surface is yellowish brown to deep reddish brown. Root bark. Both the surfaces are of identical color, deep reddish brown. Odor. It has slight and characteristic odor. Taste, intensely bitter and somewhat astringent. Then size: CINCHONA calycea 12 to 25 mm in diameter, CINCHONA legergenia 12 to 25 mm in diameter, CINCHONA officinalis up to 12 mm in diameter and 1.5 mm in thickness, CINCHONA succirubra 20 to 40 mm in diameter and 2 to 5 mm in thickness. Shape: either in the form of quills or curved pieces. The different varieties can be differentiated as under: CINCHONA calycea, broad longitudinal fissures with transverse cracks 6 to 12 mm apart. CINCHONA legergenia, similar to CINCHONA calycea, cracks are more numerous and less deep. Some pieces show longitudinal wrinkles and reddish warts. CINCHONA officinalis, transverse cracks numerous, less than 6 mm apart. CINCHONA succirubra, well marked longitudinal wrinkles, relatively few transverse cracks. Some pieces show reddish warts. Next point: microscopic characters. CINCHONA exhibits the typical histological characters of the bark. The cork cells are thin walled, followed by phylloderm. The cortex consists of several secretory channels and phloem fibers. Medullary rays with radially arranged cells are present. Idioblast of calcium oxalate is the specific characteristics of CINCHONA bark. Starch grains are present in the parenchymatous tissue. Stone cells are rarely present in the structure. A few of the cork cells are lignified. Medullary rays are 2 to 3 cells wide. Chemical constituent: it contains about 25 alkaloids in bracket 5 to 10% bracket complete, mainly quinine, quinidine, cinchonine, cinchonidine. It also contains bitter glycoside, quinolone, cinchofulvic, cinchotanic and quinic acids. Red coloring matter: CINCHONA red, tannin, starch. The other alkaloids are hydroquinone, hydrocinchonidine, quinicine, cinchonicine. Next point: chemical test. First one: heat the powder bark in a dry test tube with little glacial acetic acid. It produces purple vapor. If treated with dilute sulfuric acid, it gives blue fluorescence. Second one: to the powdered drug, add bromine water and ammonia and heat. Emerald green color due to formation of thylaquine is produced. Next point: uses. First one: it is used as antimalarials and antipyretic. Second one: it is a bitter tonic and stomachic. Third one: cinchonidine is used in rheumatisms and neuralgia. Fourth one: quinidine is used as cardiac depressant. Next point: substitutes of CINCHONA. First one: Cuprea bark. It is dried bark obtained from the stems of Remigia. Pendiculata, family Rubiaceae. It is obtained from the central and southern Colombia. It is coppery red color bark with granular and splendidy fractures. It is hard, compact and heavy. Histologically, it contains numerous stone cells. It contains small amount of alkaloids as compared to cinchona. The alkaloids are quinine, quinidine, cinchonamine and cuprin. Second one, Fells cuprea bark. It is obtained from Remgia puridinia. It contains no quinine but traces of cinchonine and cinchonamine. Additionally, it also contains an alkaloid known as cusconidine. Next point, shape of bark. The shape or forms of the bark is dependent upon the method adapted for its preparation, type of incisions and extent of any shrinkage of tissues. First one, when the bark is removed from the large trees and dried under pressure, the flats are produced. Example, Quillia arjuna. Second one, when the bark is removed from the small branches due to shrinkage of the tissue, it tends to curve, forming the concavity on the inner side, produce the curved pieces. For example, wild cherry, cassia. Third one, if the concavity is on the outer side of the bark, it is described as recovered. Example, kurchi. Fourth one, when the shrinkage of the tissue is the extent and it forms deep through or channels, it called as channel. Examples, ashoka, cinchona, cassia. Fifth one, if one edge of bark covers the quill's shapes, it is called as quilled bark. Example, cascara, cinnamon. Sixth one, if both the edges of the bark roll independently forming the quill, it is described as double quill. Examples, Java cinnamon. Seventh one, if one quill of bark is put inside other quill to form a compound quill, for example, cinnamon. Thank you.